Welcome to our computational biology lecture on measures of variability. Today, we are going to explore why simply knowing the average of a dataset is never enough to truly understand it. We will learn how to measure the spread, noise, and reliability hidden within biological data, building your intuition from the ground up so you can analyze experiments with confidence. Let us start by imagining two biology classes. In Class A, every single student scores exactly 75%. In Class B, half the students score 50% and the other half score 100%. Both classes have the exact same average of 75%, but the situations are completely different. This shows us why the mean is never enough. We need a way to measure how spread out or scattered the data points are. In computational biology, this is critical. If a new drug lowers blood pressure by an average of 10 points, but causes wild fluctuations in individual patients, it could be dangerous. Variability tells us the true story. Now that we understand why we need to measure spread, how do we actually calculate it? A naive approach would be to just add up the distances of every point from the mean. But because some points are above the mean and some are below, the positive and negative distances would perfectly cancel each other out, giving us 0. To fix this, we square each distance before adding them up, turning all numbers positive. Then we find the average of these squared distances. This fundamental concept is called variance. Because we square the differences, variance gives much more weight to extreme outliers, meaning a data point that is far from the mean will increase the variance significantly more than a point that is close. This brings us to the mathematical formula for sample variance. Let us break it down. The symbol S squared represents the sample variance. Inside the numerator, we take each individual data point, xi, subtract the sample mean, x bar, and square the result. The Greek letter sigma tells us to add all these squared deviations together. Finally, we divide by n minus 1, where n is the sample size. We use n minus 1 instead of just n to correct for a slight mathematical bias, giving us a better estimate of the true population variance. Notice that if the data spreads out further, the numerator grows and the variance increases. While variance is mathematically beautiful, it has a major practical flaw. The units are squared. If you are measuring the weight of mice in grams, the variance gives you a number in squared grams, which makes absolutely no physical sense. To fix this, we simply take the square root of the variance. This brings our measurement of spread back into the exact same units as our original data. This new, highly practical number is called the standard deviation. It is the most popular and widely used measure of variability in all of science because it is so easy to interpret. Here is the formula for standard deviation. As you can see, everything inside the square root is the exact same formula we just learned for sample variance. We are simply taking the square root of the entire calculation. The symbol S represents the sample standard deviation. By taking the square root, we reverse the squaring we did earlier, returning the metric to the original units, like grams or meters. If the data points are tightly clustered around the mean, the differences are small, making the standard deviation very small. If they are spread out, the standard deviation grows. Before we move on, let us see this with a concrete example. A biologist measures enzyme activity in 5 cell cultures, getting values of 10, 12, 15, 18, and 20 units per liter. First, we find the mean by adding them up and dividing by 5, which gives us 15. Next, we subtract this mean from each data point and square the results to get our squared deviations, which sum to 68. We then divide this sum by n minus 1, which is 4, giving a variance of 17. Finally, we take the square root of 17 to find our standard deviation. The final answer is approximately 4.12 units per liter, meaning the enzyme activity typically deviates from the mean by this amount. Now that we have calculated standard deviation, let us visualize what it actually means. You are looking at a normal distribution, often called a bell curve. The horizontal axis represents our data values, and the vertical axis shows how frequently they occur. The peak in the center is the mean. The standard deviation, represented by the Greek letter sigma here, acts as a measuring stick. In any normal distribution, approximately 68% of the data falls within one standard deviation of the mean, shown in the shaded blue region. Furthermore, 95% falls within 2 standard deviations. This predictable property makes it an incredibly powerful tool for biologists to identify normal versus abnormal traits. This brings us to another crucial concept. Imagine you want to know the average height of all penguins in Antarctica. You cannot measure every single penguin, so you measure a sample of 50. You calculate the mean, but how confident are you that it matches the true population mean? If you took another sample, you would get a slightly different average. The standard error of the mean measures exactly this uncertainty, while standard deviation tells you how spread out the individual penguins are. The standard error tells you how much the sample mean itself would jump around if you repeated the experiment over and over. Let us look at how we calculate this uncertainty. The formula for the standard error of the mean, or SEM, is the sample standard deviation, S, divided by the square root of the sample size, n. This formula tells us 2 very important things. First, if the natural variability of the data is high, the standard error will also be high. Second, and most importantly, as you increase your sample size n, the denominator grows larger, which shrinks the standard error. This mathematically proves why larger sample sizes give more precise and reliable estimates in biological experiments. Let us apply this to a real scenario. A researcher measures the expression level of a specific gene in 16 different tumor samples. The sample mean is 120 units, and the standard deviation is 32 units. We want to know the precision of this average. We take our standard deviation of 32 and divide it by the square root of our sample size, 16. The square root of 16 is exactly 4. Dividing 32 by 4 gives us a standard error of 8 units. This means that if we were to repeat this experiment with different batches of 16 tumors, our calculated mean would typically fluctuate by about 8 units from the true overall average. Now that we have measured absolute spread and precision, we face a new problem. Suppose you are comparing the variability of 2 completely different things, the weight of elephants and the weight of mice. An elephant's weight might vary by 100 kilograms, while a mouse varies by only 5 grams. If you just look at the standard deviation, the elephant's variability looks massively larger. But 100 kilograms is a tiny fraction of an elephant's total weight, whereas 5 grams is a huge percentage of a mouse's weight. To fairly compare datasets with vastly different scales or units, we need a relative measure. This is exactly what the coefficient of variation does. Here is how we calculate this relative measure. The coefficient of variation, or CV, is defined as the ratio of the standard deviation, S, to the mean, x bar, multiplied by 100. Because we divide the standard deviation by the mean, the original units completely cancel out, making it a dimensionless number. Multiplying by 100 simply converts this decimal fraction into a readable percentage. If the standard deviation increases while the mean stays the same, the CV increases, indicating higher relative variability. This property makes it incredibly useful in computational biology for comparing the degree of variation from one data series to another, even if they are measured in completely different units.
Let us see this in action. A biologist studies cells with a mean diameter of 50 micrometers and a standard deviation of 5. Inside, a protein has a mean mass of 2,000 Daltons with a standard deviation of 400, which has higher relative variability. We cannot compare micrometers to Daltons directly. For the cell diameter, we divide 5 by 50, giving 0.10, or 10%. For the protein mass, we divide 400 by 2,000, giving 0.20, or 20%. Even though the absolute standard deviation of the protein mass was numerically much larger, calculating the percentage proves that the protein mass is also relatively more variable compared to its own mean. It is twice as variable. We have covered a lot of ground today. Let us summarize the key takeaways. We learned that variability is essential because it tells us how spread out our biological data is, revealing the true noise and diversity. Variance provides the mathematical foundation by averaging squared deviations. But standard deviation is the most practical tool because it shares the same units as the original data. When we need to know how reliable our sample mean is, we use the standard error. And finally, when we need to compare the spread of completely different datasets, we rely on the coefficient of variation. Mastering these concepts allows you to truly understand the significance hidden within complex biological datasets.